Is it okay for everyone if we record? Because if you are not okay with recording, please tell me in the chat so I stop recording. We will not be diffusing the recording. It's only for internal use. But if you are not okay with the recording, please tell me in the chat because I need the agreement of everyone to record the session.
I agree.
Thank you. Thank you, Steve.
J'ai un peu un chat dans la gorge. Si jamais je dois m'arrêter, je couperai la caméra et le micro.
Oui, il n'y a pas de souci. Il n'y a pas de souci. Sentez-vous libre. Je vais attendre juste jusqu'à 5h05 et puis on commence. Voilà, on est déjà à 60. Ce sujet, il est vraiment un sujet d'actualité.
Oui, et comme je disais, je pense qu'il faut vraiment informer. Je suis ravi que les gens s'y intéressent et qu'ils y passent un peu de temps pour comprendre comment les choses fonctionnent et justement pouvoir choisir ce qu'ils veulent. Il n'y a pas à imposer les choses, on peut s'informer et choisir ce en quoi on croit.
They are asking me if I can send the recording to the participants of the webinar. So I need the agreement of Dr. Pascolo. If he says okay, we can send the recording.
Yeah, it's okay.
Okay, so we can send the recording. Nathalie, Laura عم بتقلي le lien ne fonctionne pas. ليش le lien ne fonctionne pas? Tu peux lui envoyer par WhatsApp. Je vais lui envoyer.
C'est le même lien que j'ai envoyé à tout le monde.
Oui, je vais lui envoyer.
Et votre organisation, donc, quelle est-elle et que fait-elle?
Alors, nous sommes à l'Université Saint-Joseph de Beyrouth et la chaire que je vais présenter, c'est la chaire d'une fondation qui s'appelle la Fondation Diane, qui a été créée par Madame Diana Fadel. Je pense qu'elle est soit parmi nous, soit qu'elle va arriver. Il y a des collègues de la fondation et c'est une fondation qui s'intéresse à toutes les questions liées au développement durable et à l'environnement, avec un incubateur, des formations. Je vais présenter tout ça.
Très bien.
Et l'Université Saint-Joseph, c'est aussi la plus grande université francophone de la région.
C'est un grand bonheur que la France ait autant de francophones, qu'il y ait autant de francophones partout dans le monde, d'avoir ces liens privilégiés. Même si on va parler en anglais,
oui,
c'est quand même quelque chose qui nous rapproche.
Participants, they have the choice to ask that question in French, in English or in Arabic. I will say it too. So the language will not be a barrier. So vous pouvez poser vos questions en français, en anglais ou en arabe, il n'y a pas de souci, et puis on y répondra. Je vais juste... Si tu peux, Nathalie, envoyer le lien alors seulement. Alors là, on va demander à tout le monde de mettre mute. Voilà. Donc, on va commencer dans 1 minute. Tu vas faire attention, Nathalie, à faire mute et Tracy à chaque fois que quelqu'un ouvre son micro, parce qu'on est nombreux.
Si jamais à un moment vous ne m'entendez pas, dites-le moi. J'ai aussi
oui.
des micros au cas où.
Oui.
Je voulais
voilà,
juste...
Laure, elle est avec
oui,
nous,
c'est bon, c'est bon. Oui, merci, merci.
Voilà, donc il est 5h05. Hello everyone. Thank you so much for being here and for being numerous to participate in our webinar. Thank you so much to Professor Steve Pascolo We have the honor to be with you in this webinar. So before starting the webinar, let me show you a very small video concerning our share. I will present myself, maybe at the beginning some people don't know me. So my name is Fadi Al-Haj. I'm the director of the share of Fondation Diane for Education, Ecocitizenship and Sustainable Development at St. Joseph University. So thank you so much for attending this webinar. We'll be presenting so as I said, the short video before presenting Professor Pascolo.
صيادلة لبنان احتجاجا على ارتفاع سعر صرف الدولار مقابل الليرة اللبنانية أكد نقيب الصيادلة.
If you can mute yourself, please. When you are not speaking, please mute yourself. Thank you. So let me introduce Professor Pascolo. Professor Steve Pascolo is trained as an immunologist at the Pasteur Institute, Paris, France. He used mouse models to test and develop mRNA-based vaccines, direct injection of mRNA, during his postdoc in Germany. In 2000, he co-founded CureVac with Dr. Hor and Dr. von der Mel. Dr. Pascolo was chief scientific officer of the company from 2000 till 2006, developing the technology, implementing the worldwide first GMP production of mRNA, and performing the worldwide first clinical studies where humans, including himself, got injections of in vitro transcribed mRNA. In 2006, he joined the oncology department of the University Hospital of Zurich, Switzerland, and continued the development of immunotherapies based on RNA. In 2008, he founded Mixture Pharma to support this work. In 2017, Dr. Pascolo implemented in Zurich an academic mRNA platform in collaboration with several research and clinical departments in Zurich. He optimizes, tests, and implements mRNA-based therapies. Thank you so much, Professor Pascolo, for being with us this evening, this afternoon, to discuss about the link between mRNA and well-being and all the challenges of this kind of vaccination. Thank you, Dr. Pascolo. For all the participants, please feel free to ask your questions in the chat box during the presentation in any language, in French, in Arabic, or in English, and we will manage to answer all your questions. Thank you.
Thank you so much. Do you hear me well?
Yes, excellent.
So thank you so much for your invitation. I'm happy to present the background on vaccines and mRNA vaccines, especially in the context of COVID-19. And I hope there will be many questions and topics we can speak about together. So I will start sharing my screen.
Excellent.
So you see well my screen?
Yes.
Perfect. So I will come back a little bit to my background. Indeed, it was usually academic and industry in parallel. In Tübingen in Germany, I worked on different types of vaccines, including peptide vaccines, adenovirus vaccines, DNA vaccines, and mRNA vaccines. And for the development of the mRNA vaccines, we co-founded CureVac in 2000. And I left the company. I have no shares of the company. I'm not an employee anymore of the company since 2006, so I have no conflict of interest. I don't benefit from the mRNA vaccines that are reaching the market. I joined fully the university in 2006, developed new immunotherapies against cancer, and to support them, I created Mischer Pharma in Zurich, which is a company just supporting patent and development of new products against cancer. And as you said, in 2016-17, I implemented the worldwide first messenger mRNA platform in a university. And through this platform, I try to stimulate people to do fundamental and preclinical and clinical research using mRNA, which is a safe alternative to many other formats of therapies, which are, for example, viruses or DNA. mRNA is a flexible and safe format, as I will show you later. And so through this platform, I can design, produce, and give to any scientist in Zurich mRNA ready to be used, explaining them also how to use it so that people can develop their preclinical or clinical research with messenger RNA. Let me start by giving a background on vaccines. So here, this table shows you a non-exhaustive list of vaccines that can prevent disease. And I group them in 3 families. The top family here are attenuated viruses. So these viruses are attenuated, but they are alive. And they are mostly injected subcutaneously or intradermal. On the contrary, all the other vaccines are inactivated, dead viruses, or subunit vaccines. So all these vaccines here, inactivated and subunits, are dead in a way. And they need to be injected most of the time intramuscular with adjuvant. So dead vaccines are injected intramuscular with adjuvant. Live vaccines can be given subcutaneous, which is preferred. And I want to highlight that because you will notice that quite many of these vaccines, which are attenuated viruses, contain RNA viruses. Rubella, measles, mumps, yellow fever, these are mRNA viruses. In other words, when you get these kind of vaccines, or when your children have gotten these kind of vaccines, which are used in the old world, they got already mRNA vaccines. So we hear now that mRNA vaccines are new. Which is new is synthetic mRNA vaccines, but natural mRNA vaccines exist already since decades, and they work here. All these viruses, all the RNA viruses, they work in these vaccines by infecting your cells, giving to your cells their mRNAs, which are then translated, trigger inflammation, and trigger an immune response. The same way that the new mRNA vaccines are doing. They all work the same way. They go from outside, deliver their mRNA in your cells, and your cells produce the proteins, make the inflammation, and stimulate the immune system. So in that sense, the mRNA vaccines are old technology. What is new is just that it's synthetic. And in the past, it was natural. But the mechanism of action, and so the safety is the same, and it has been proven since many years to be efficacious and safe mRNA vaccines. I illustrate that in more details here. These are the mumps, measles, rubella viruses in the vaccine. They are all attenuated using different methods, either culture in animal cells or lower temperature, so to make viruses which can still live and infect, but but do not give the disease. And all these viruses, they have complex structures. They have all one or few mRNAs, positive or negative strands, and they contain a lot of proteins. They contain a lipid bilayer containing also some other proteins of a virus. So each of these viruses has RNA and has many other components, including also components coming from the production. So all these vaccines are produced on animal cells. You need animal cells to produce these viruses. It can be cells in cultures, or it can be fertilized eggs from chicken. And then you need to purify these attenuated viruses from the cultures or from the eggs. And for this reason, you also have in these vaccines impurities, a lot of impurities coming from the cell culture or coming from the eggs, which are a concern when you are allergic. So these complex viruses contain, in addition, some reagents from the cell culture. On the contrary, here I depict the new kind of mRNA vaccine, the synthetic. IVT means in vitro transcribed. I will come back to that later. Synthetic mRNA vaccine. They contain one mRNA molecule coding for one protein. All these viruses here, they code for many proteins. Here, in the synthetic mRNA vaccine, the mRNA codes one protein and is encapsulated in a liposome consisting of 4 lipids. So in the synthetic mRNA vaccines, you have 5 components, mRNA and 4 lipids. It's very pure, and easy to produce, as opposed to the others, which have containing many things and are not so easy to produce. Going to COVID, there are now 13 vaccines that have been approved in the world. 2 are based on subunit vaccines, proteins, purified and produced in vitro and purified. 2 are this mRNA vaccine, on which I will focus. 5 are recombinant adenovirus vaccines, so they are comparable to attenuated viruses. And 4 are inactivated SARS-CoV. So these 4 here are similar to the flu vaccine. The virus is produced in vitro. It's inactivated with a chemical like formaldehyde and used as a vaccine. So you have all these families now available against COVID: inactivated, attenuated, mRNA, protein. So I think, Fadi, you have a question.
Yes, we'll be launching a poll here. So we want to ask you a question to have an idea about what do you think about these vaccines. So the question is, if you have the opportunity to choose your COVID-19 vaccine, which one would you choose? Pfizer, AstraZeneca, Johnson & Johnson, Sputnik, Sinopharm, or none? So it would be great if you can vote, all of you, to have just an idea of your opinion concerning vaccination. Yes, we have already 40 answers, over 91. So Salim is saying anyone, okay, because we need to choose one, Salim, if it's possible, just to have an idea. Excellent, so we have already 72 answers. Come on, let's wait for a few seconds more. Hadi is asking about Moderna. We'll be talking about it later on, maybe.
Maybe I can add something to give time to the people, is what I wrote here. These vaccines, they don't code for the exact same protein. All these recombinant vaccines here, they give protection against the spike of the SARS-CoV, which is the surface protein, the one recognized by neutralizing antibodies. And there are 2 forms of the spike. There is a form which is called PP, it's a modified spike, and there is the wild type spike. There are some vaccines that code for the wild type spike and some vaccines that code for the mutated spike. So they don't give exactly the same immune response, and it's important when we speak about the efficacy of a different vaccine.
So we have the answers here, if you'd like to comment, Steve.
So I'm glad to see that the Pfizer seems to be the winner, because that's an mRNA vaccine from BioNTech, actually. And as we will see, it's one that shows high efficacy, high safety, and which is broadly used in the world and provides some excellent protection. And of course, the Johnson & Johnson is one single injection, so it has an advantage. The Sputnik V, as we will see, it's attenuated viruses. It contains some safety issues, potentially, we will speak about. And some people don't want vaccines, and I can understand. And I hope that after the talk, maybe they will eventually consider to go for a vaccine, but it will be very interesting to have the feedback of the audience at the end of my talk.
Okay.
So actually,
there's a question from Maria. What are the first two vaccines technology in this slide?
These are recombinant proteins. So these are proteins expressed in vitro, purified, and made with an adjuvant in a vaccine. It's similar to hepatitis B. The hepatitis B vaccine on the market is also a recombinant protein. So if there are no more questions, I will continue. Do you hear me, or did the connection stop?
No, we can hear you.
Okay. So I will continue with actually an answer on the poll. I have the feeling that the poll has blocked my computer. Now it goes. So here I give you a subjective view of the advantages and the disadvantages of the different vaccine platforms. Again, the platforms are synthetic peptide, plasmid DNA, recombinant viral vectors, so attenuated viruses like adenoviruses, in vitro transcribed mRNA, subunit vaccines like proteins, purified proteins, inactivated viruses, and attenuated viruses. In blue are all the formats approved against COVID-19. And I show you here some steps about these vaccines. So first, their design. It's easy to design an mRNA vaccine, and this is important because if we have new variants of SARS-CoV-2, if we have a SARS-CoV-3, it will be very easy to design an mRNA coding for the protein of a new variant. When you work with viruses, it's a bit harder to know how you can modify the virus in order for it to protect against a new variant. So the speed of designing an mRNA vaccine is very fast. The upscaling is an important issue. We see here when the world needs billions of doses, it's important to upscale the production. And upscaling production of, for example, adenovirus is not that easy. You have heard that AstraZeneca has problems. The problem is that viruses are live. When you produce them in cell culture. So from one batch of thousands of liters of cell culture to another batch, you may have different quality of viruses. You may have more dead virus, more live virus, more debris, more impurities. So to have a standardized production of recombinant viruses is hard. For the mRNA, as I will show you after, it's linear and it's much easier to produce small amount and big amounts of mRNA. It requires the same condition and it's robust. Reusing established pharmaceutical conditions. Again, if you know how to produce, let's say, yellow fever virus in order to make a yellow fever vaccine, and now you want to produce a SARS-CoV-2 virus, you will have to test everything again. You will have to find the right cell type. You will have to find the right conditions of culture, the incubation time. You will have to evaluate all the methods and to redo the methods for each new virus against which you would want to vaccinate. And I will show you that this is not the case with mRNA. And the most important is here, and it comes to the poll we had just before, is the theoretical safety. I want to insist on theoretical. All the approved vaccines are safe. That's why they have been approved. They have gone through phase 1, phase 2, phase 3 clinical study. The side effects have been scrutinized, and if the vaccines are approved, they're safe. So from that point of view, one can rely on all the approved vaccines to be safe, to give side effects, and things can be always reevaluated and followed in so-called phase 4 studies. But of course, if they are approved, they are safe. Now I'm speaking about theoretical safety. The theoretical safety of adenovirus, the issue is that these adenoviruses, I will not speak much about in the rest of my talk, but I will describe it here. For example, the AstraZeneca vaccine is a combination of 5 different genomes. It contains the backbone, which is from an adenovirus of a chimpanzee. Inside of it, it was put a part of a cytomegalovirus, a promoter. Then it was put a short part of a mammalian genes, which codes for a leader sequence of a protein. Then you have 4 kilobase of a cDNA, of a copy DNA from the SARS-CoV spike. And this DNA does not exist in nature. The SARS-CoV is an RNA virus. So in the laboratory, from the RNA virus was made a DNA coding from the spike. And this DNA is put in the adenovirus from AstraZeneca. So this sequence is not existing in nature. And after this gene, there is a part coming from a cow gene for stopping transcription. So you have 5 different DNA parts which have been assembled in the adenovirus vaccine of AstraZeneca. So on this, for this reason, it's hard to predict how this construct will behave on the long run. We all know that DNA is stable. These adenoviruses are DNA. So when people get these vaccines, they get injected with a DNA containing 5 elements for which we don't really know if they can persist, if they can recombine with other adenoviruses that one gets in winter, or if they can integrate in our cells at the site of injection and eventually trigger transformation of cells. These are things that are theoretical. We will have to follow. They will be, of course, very rare, if at all happening, but that's a concern which do not exist with the other formats, because as we will see, mRNA, proteins, and dead viruses are all eliminated quickly and naturally by the body. There is no reason to think that any of this would persist in the organism. On the contrary, in theory, the adenoviruses could persist at the site of injection and give issues in many years. We don't know. I hope it's not the case, but it's a theoretical concern at this stage. So I will now focus on the 3 formats which have been approved around the world, and Novavax is not yet approved, but it will come soon. So it's purified protein, recombinant adenovirus, and IVT mRNA. So inactivated viruses are used in China and India, but so far, as far as I know, they are not approved in Europe or the US and in Lebanon. So these 3 platforms give 5 vaccines, because there are 3 vaccines on the IVT mRNA. And as I said at the beginning, all these vaccines, they protect, they trigger an immune response against the spike of the SARS-CoV-2, so the protein at the surface. It's a big protein. And some vaccines, like the one from AstraZeneca, use the wild type sequence of the spike, so the normal sequence of the spike protein, which is in the RNA virus SARS-CoV. It has been translated, reverse transcribed into a DNA, but it's the wild type protein. On the contrary, the mRNA vaccines, all the three, the two that have been approved and the one in development, use a so-called PP spike. And the PP spike was described already for the MERS of 2012 and for the SARS-CoV-1 of 2002. People who studied the spike proteins from these coronaviruses, they could show that the protein has two different structures. It's depicted here. So when you have a spike protein purified in vitro and you do electron microscopy, you can see two shapes of the spike protein, an elongated one and a mushroom kind of shape. So the protein can fold in two ways, the wild type protein. And the mushroom form is the form at the surface of a virus. So when you vaccinate, you want to use the mushroom form. You want to get antibodies recognizing this form because this is the one at the surface of the virus. Antibodies recognizing this form will not recognize the virus. So it's important to vaccinate with a protein that has this form. And this form of the mushroom is stabilized when two prolines are replacing amino acids in position 986 and 987. So these two amino acids are replaced by proline, and that is sufficient to give to all spike protein the mushroom shape, which is called the pre-fusion conformation. And this is true for MERS. It's also true for SARS-CoV. So at the beginning of the SARS-CoV-2 pandemics, the companies have decided whether they use the wild type or the prefusion stabilized form. And most of the companies have decided to go for the prefusion stabilized form, not AstraZeneca. So that could also explain differences in antigen, let's say in the protection triggered by these different vaccines against variants, because they don't code for the exact same protein, which could have consequences on the repertoire of antibodies and their capacity to recognize different variants. So the efficacy of these 5 vaccines is really high. It's above 60, it's most of the time 90% against the SARS-CoV-2 of 2020. Now, as you know, there are many variants. The most scary one is the one identified in South Africa. And it is known that the mRNA vaccines, they give antibodies which protect against the South African variant, even if the protection is a bit lower, but it still protects. The AstraZeneca vaccine seems not to protect against the South African variant. It might be due to the fact that the AstraZeneca vaccine codes for the wild type protein, which can have the 2 structures, while the mRNA vaccine code for the proteins, which has only the prefusion shape. The dosing of these 5 vaccines is indicated here. It's always around micrograms, 5 micrograms, 2 micrograms, 30 micrograms, 12 or 100 micrograms. So that's more or less the dose. It's very low. If you think about here is the highest dose is 100 microgram. So it's like 1,000 times smaller than an aspirin tablet, what is injected. So it's a very tiny amount of product which is injected in vaccines. And all of them are injected twice with 3 to 4 weeks interval, except the Johnson & Johnson, which I did not depict here, but the Johnson & Johnson is similar to the AstraZeneca. It's an adenovirus. However, it doesn't code for the wild type protein. It codes for the PP, so for the prefusion stabilized protein, and it's given only once. Now, the theoretical concerns, again, here, when you use a protein vaccine, the purified protein, one concern is are contaminants. So in proteins, when you purify your proteins, you always have contaminants. And these contaminants, including proteins which are the wrong structure, could trigger an immune response, which is inefficacious. So that's a concern of protein vaccines. The concern in adenovirus, recombinant adenovirus vaccines is the risk of recombination, integration in the genome. as I mentioned before. There are also other risks of these adenoviruses not being accepted by the public. One thing I didn't say before is that they are produced on human cells, these adenoviruses. The one of AstraZeneca is produced by HEK cells. HEK cells are human embryonic kidney cells. So this is a cell line that was made in 1973 from an abortion, and the cells from the aborted fetus were cultivated in vitro, transformed with an adenovirus, and that's why they can produce deficient adenoviruses against SARS-CoV-2 when the SARS-CoV-2 virus, when the adenovirus coding for SARS-CoV-2 infects these cells can somehow complement the deficiency in the adenovirus that is used for the vaccine and produce viruses. And so for religious reasons, some people don't want this vaccine, and also the production is a bit complicated for this vaccine. So here are the pros and cons of the different vaccines that will be available to all of us, that are available, some of them, and will be available. Some questions on this precise slide?
Yes, we have questions here, and maybe we can ask our WooClap question concerning the, I will add a link in the chat box. So if you copy this link, or if you click on it, maybe Natalie will put it as a clickable link now in the chat box. You can click on it, and then we'll see your answers. I will be asking you, Steve, if it's possible to stop sharing, because we'll answer this question, and then we go back then to your slides. Thank you. So, so I'm asking you to, Natalie, can you add this link in a clickable way in the chat box, please? Otherwise, you can copy it. Natalie just added. You just click on it and answer, either on your cell phone or you just click on the link. So I will be sharing the results right now. So can you see my slide?
No, I see the chat.
No. Okay, I will share it again. Okay. Maybe now you can see it. Can you see it now?
Yes.
So what are your concerns about COVID-19 vaccine safety? We can see the answers. So long-term side effects. I will be waiting for a few seconds, and then we will try to analyze the answers. So you can click on the link or copy the link on your cell phone or on the browser and answer the question, all of you. So we have until now like 25 participants. We need more answers. What are your concerns about COVID-19 vaccine safety? So we have these answers. We have side effects. We have long-term side effects. We have mutations, specific allergies. Okay, thank you. So maybe we can stop answering to analyze the answers.
Yes, so side effects is important. Vaccines have side effects. If you have a vaccine that gives you no side effect, it's a vaccine that doesn't work. I mean, in a large population. So if you vaccinate millions of people and you have no side effect, the vaccine doesn't work. If a vaccine works, you will expect side effects in some people. And because that's the way the vaccine works, it activates your immune system. So you get a little bit of fever, headache, and sleepiness. So some people don't notice anything, some people notice it more, but you expect from any vaccine that works to have these side effects. So it's right to mention side effects. The vaccines that have been approved against COVID, they do have side effects, but most of the time they are mild, and you cannot, it's like for the flu vaccine or for any other vaccine, you may have some people who have more side effects, and that's something to keep in mind. And I think the campaigns are being done carefully, where the people who get vaccinated, they have first to answer a few questions about their potential allergies or medications that they may take, which may be not compatible with the vaccination. So it's important to keep in mind side effects. There are side effects, and if someone has a special chronic condition, before getting injected by a vaccine, she should say to the personnel, to the medical personnel about these conditions to make sure that there is no contraindication, because this condition could be then really a bad combination with a potential side effect of the vaccine.
So we have some question in the chat box. Maya is asking, what about Pfizer side effects after 4 months? Why we should give it to a normal person who is vulnerable, not vulnerable? So is there any side effects 4 months concerning Pfizer?
Not as far as I know. The side effects are always transient and resolve on their own. On the contrary, the virus infection has long-term side effects, even in younger people. So the SARS-CoV-2 infection has, in many people, as we know, is deadly, and on many people has long-term side effects. So that's clear, but I didn't hear about long-term side effects from a vaccine.
Okay, so Roy is asking, an asthma, lungs problem person can take the vaccine?
There is no contraindication. I mean, asthma is not a contraindication for vaccines. So that's the same for flu vaccines or for any other vaccines. Autoimmunities and other kind of chronic disease are usually no contraindication for vaccines. Now it can be case by case, in case someone has strong asthma or strong allergies. One has to be careful and to really review which vaccine he will get and where are the risks. So there might be some people who would rather maybe not take the vaccine, but in general, asthma and other type of chronic disease, like also autoimmune disease, are no exclusion criteria for getting the vaccine.
Okay. Romy is asking, can Pfizer be potentially the cause of embolie for a senior person with Alzheimer's?
Single cases are always difficult to analyze. So there have been embolies in people always. So that's not something that is new. You have cases of embolies everywhere, every day. And actually, embolies are triggered by the SARS-CoV infection. So now there's a clear link between pulmonary embolies and SARS-CoV infections. And that was the case of my mother, who had a lung embolie early in 2020. And that's a known now established link between infection with SARS-CoV-2 and lung embolies. So there are much more risk of getting a lung embolie from the SARS-CoV-2 infection than from any vaccines that have been approved, because any vaccine that has been approved has not yet shown a link to embolie. There could be some cases that have to be studied, whether there is a link or no, but as far as I know, from the hundreds of millions of people who have been vaccinated and tens of millions with the mRNA vaccines, for example, against COVID, there was no proof that this vaccine gives more embolies. But again, the SARS-CoV-2 virus gives embolies.
Okay, maybe we can take one more question and then we continue the presentation and then we answer the other questions. But there are too many questions in the chat box, which is very interesting. So I will ask you one more question. We continue and then we come back to the questions. So Christine is asking, is there a way to test the hypersensitivity to the PEJ?
To the PDJ? What is PDJ?
PEG. Is there any way to test percentage?
to the PEG, there could be some allergies against PEG. So PEG is at the surface of the liposomes that encapsulate the mRNA. And there have been people having quite severe allergies in Great Britain after getting injection of the BioNTech Pfizer vaccine. And it was probably due to the PEG, to allergy to PEG. So that again comes to highly allergic people. So these people in Great Britain who had severe allergy after injection of the Pfizer BioNTech, these are people who know that they have severe allergies. These people have always their tablets with them, their medications with them, because they know they have severe allergies. So it's a recommendation if somebody has very severe allergies to really speak with his medical personnel before getting the vaccine, and in all cases to have his medication with him. So people who have severe allergies should be careful.
Yes, just one more question, because Carol is saying, please answer this question. So what about those who have immune deficiency and immunity problems?
Again, that's not a contraindication of vaccines. So usually if the person who has a deficiency gets routine vaccinations against influenza and against other viruses, then this person should get the vaccine. The problem in these persons is that the vaccine would not work too well, but in these persons, the infection with the SARS-CoV would be very problematic. So people with immunodeficiencies should be vaccinated. Again, one needs to speak with his physician who knows exactly the case and who knows the background and who knows if normal vaccination work and are safe. In that case, then the SARS-CoV-2, the COVID vaccine should be given. But in general, these people are at risk to the infection, so they should be actually vaccinated, yes.
Okay, so we can continue. We can share your PowerPoint if you want and continue, Steve, and then we'll stop later on to answer some more questions.
So I will now focus on the mRNA vaccines. Sorry, there is always a delay when we come back to the presentation. The computer needs some time to react and to go to the next slide.
Yes, actually we see, yes.
But it's always the same slide. I'm trying to go to the next one, but it does not respond. Maybe I need to escape from the presentation.
Okay.
Because now, it's okay. So I will focus on mRNA. So, and mRNA vaccines. So I remind you what is mRNA. So here is a cell, a mammalian cell, for example, a eukaryote. You have here a nucleus. In the nucleus, you have the DNA. Parts of the DNA are transcribed into pre-messenger RNA. And then this molecule, the pre-messenger mRNA, is spliced, is modified, is capped at the 5'. So there is the creation of this structure at the 5' as a reverted guanosine with methyls that is linked with 3-phosphate to the first nucleotide, which is normally also methylated. So this is the so-called cap structure, which is specific for messenger RNA. And the 3' end is polyadenylated. This mRNA goes out of the nucleus, is further modified. There are some methylations in the mRNA. There are some conversion of some base, uracil to pseudouridine, or guanosine to inosine. So there are several, adenosine to inosine. So there are several modifications on the mature mRNA, which is then translated into a protein. The advantage of using mRNA for therapies is that it's transient. As I said at the beginning, mRNA and proteins are transient, while DNA is stable. It's possible to produce in vitro the mature messenger RNA. And you can do the production in 2 ways. One called non-modified mRNA and one called modified mRNA. The non-modified mRNA is a synthesis of mRNA containing the 4 non-modified nucleotides, SCGU. The modified mRNA is made in conditions where you replace the U by a pseudo-U. And the difference of these two mRNA is depicted here. The non-modified mRNA, when you inject it, it's recognized by cells of the immune system because cells of the immune system have toll-like receptors, and these receptors in the endosomes, they recognize nucleic acids. So they recognize RNA. So when you inject a non-modified mRNA, some cells of the immune system will detect it and will immediately trigger inflammation, which is a good thing for a vaccine. So when you vaccinate, when you want to vaccinate, you produce messenger RNA in vitro that is non-modified. And there are many vaccines that are in development based on mRNA. So there are anti-cancer vaccines, antivirus, anti-allergy, and of course, in antivirus, we have now the approved anti-COVID mRNA vaccines from Moderna and from BioNTech Pfizer. Another way to do mRNA, as I said, is to have it modified so that it's not recognized by the immune receptors. So when you inject modified mRNA, which was the strategy from Moderna, Moderna stands for modified RNA. And when you inject this mRNA, you don't get an immune response. So that is what was said at least in 2005, when in Pennsylvania, Drew Weissman and Cathy Carrico have described this modification. It was said that then the mRNA does not trigger an inflammation and does not trigger an immune response, the opposite of vaccine. So this modified mRNA was then used by Moderna to develop gene therapies, like expression of therapeutic proteins. They work on expressing erythropoietin using mRNA coding for erythropoietin, which is injected, or mRNA coding for antibodies or for bispecific antibodies. So you can, thanks to the pseudouridine modification, make mRNA that you can inject, will not make an immune response, but will code for the protein that is the therapeutic protein of interest. You can also use this modified mRNA to reprogram cells to make, for example, stem cells, or to change the genome using mRNA coding for meganucleases or for Cas9. Surprisingly, in 2017, the team in Pennsylvania of Drew Weissman has shown that modified mRNA can be used for vaccination. And this is counterintuitive because since 2005, the modified mRNA was said not to induce an immune response. It was the exact opposite of a vaccine. So it came as a surprise in 2017 that modified mRNA can be used for vaccination. It's still not explained how this modified mRNA works in vaccine, where is coming the inflammation, since the mRNA is not inflammatory. So that's still questions which are not answered. And importantly, contrarily to what is said in the world, there is for the time being no evidence to show what is best, whether it's non-modified or modified RNA for vaccination. So we don't know. So far, there was no comparison well done to compare non-modified to modified mRNA for vaccination. In the last 15 years, most all vaccinations have been done with non-modified, and it's only very recent, as I said, since 2017, that the modified mRNA is used in vaccines. So we are still waiting to know what's best to vaccinate. Is it non-modified or modified? It probably depends on the formulation and the site of injection. But a general picture is that non-modified mRNA is good for vaccination and modified RNA is good for gene therapy. Here is how we produce mRNA in vitro. You need only a few reagents. You need a plasmid DNA depicted here, which contains the gene of interest. In the case of COVID, this gene is the spike, is coding for the spike protein of SARS-CoV-2. And the first step of the production is to linearize this DNA with a restriction enzyme that cuts after the gene. So this takes a few hours and is very robust. It works with any plasmid, whether your plasmid contains a gene coding for the spike of SARS-CoV-2 or for another protein of cancer, for example. Once you have a linearized plasmid, you put the RNA polymerase and the nucleotides and the cap analog And then the enzyme, the RNA polymerase, will produce mRNA from this part, like a photocopy machine will copy a book. The book is the DNA, the photocopy is the mRNA, and the photocopy machine is the RNA polymerase. So the RNA polymerase will copy the DNA. From one molecule of DNA, the RNA polymerase will make 1,000 molecules of messenger RNA within few hours. So it's very efficient. It's again like a photocopy. You press a button and your DNA is photocopied into many, many mRNAs. And this takes, again, a few hours. And again, it's robust. That means it also works independently of the sequence of the gene. So once you have it running, you can use the transcription for any gene. It's always working. After transcription is completed, you put the DNase in the reaction, and then the DNA is destroyed. And at the end, you just need to purify the mRNA. You can purify it by precipitation with lithium. There are some more advanced ways to purify it better, but that's the last step, basically, purification of the mRNA. So that's a quick process. Here are the timelines in the laboratory. Of course, when you produce it pharmaceutically, between each step, you do controls. And at the end here, you do also a lot of controls like sterility that take more time. Of course, here are the timelines only for the production in the laboratory. But similar timelines are true for pharmaceutical production, except that in between each step, you need to do some controls which take a few days. And that's why for pharmaceutical production, this process can take 1 month. A few facts on mRNA, which I want to highlight because people have wrong information about RNA or prejudice. First, mRNA is very stable. People think mRNA is not stable. It's not true. mRNA is very stable in the absence of RNases. It's the same for DNA. If you have DNA and you put DNase, the DNA will be destroyed. If you have protein and you put protease, the protein will be destroyed. So RNA is very stable, but it has to be free of RNases, of course. And the fact that RNA is very stable was recognized by the EU, the European Union, who gave in 2014 to CureVac a 2 million prize for vaccine that do not need the cold chain. So CureVac could show that mRNA is very stable at room temperature when it's RNA free, and for that reason, they got this prize of the EU. Actually, I would even go further. RNA is the most stable biological molecule. It's the only one that you can heat to 95 degrees. So any biological molecule, protein, DNA, viruses, if you put them to 95 degrees, they will be degraded and denatured. mRNA, you can put it to 95 degrees, nothing happens. You can freeze it, lyophilize it, resuspend it, heat it up. It will always be functional. And actually, at the end of the GMP production, of the pharmaceutical production that I made in CureVac, I had a step of 95 degrees, 5 minutes, to make sure that there would be no possible viruses in this preparation, should there be some viruses in any component of the production. So it's the only biological molecule you can heat to 95 degrees. So in that sense, mRNA is actually the most stable biological molecule. However, the liposomes that contain the mRNA in the vaccine, they may not be stable. So that's why the vaccines that are against COVID with mRNA are kept frozen. It's not because of the RNA, it's because of the liposome around the RNA. The liposomes are lipids, and at room temperature, they make vesicles which may change size or aggregate or fuse together, so that will change their functionality. So because of the liposomes, the mRNA vaccines at the moment are stored frozen. It's not because of the RNA. The RNA itself is very stable at room temperature, but the liposomal formulation may need to be frozen. CureVac has a formulation they claim to be stable at 4 degrees, but the Moderna and BioNTech formulation so far, they have to be stored at -20, and they are stable a few days at 4 degrees, which is good enough. Now another point is that before 2020, RNA in liposome was already an approved drug. It was not mRNA, it was siRNA, small inhibitory RNA. So there is a drug that has been approved in 2018, it's called Onpattro, and it's a liposomal formulation of a siRNA, which can treat patients suffering from hereditary transthyretin-mediated amyloidosis. So this is a genetic disease, and this disease, since 2018, is treated by a liposomal formulation of siRNA, which is given at 30 milligrams intravenous every 3 weeks. You will see later that the vaccine is given at 30 micrograms, 1,000 times less than Onpattro. It's just for you to realize that there are already patients getting RNA in liposomes, but at doses 1,000 times higher than the dose that we get for vaccines, and their whole life, every 3 weeks, these patients get Onpattro injection intravenous. One more point is that IVT mRNA vaccines are vegan. As I showed you on the previous slide, the production uses enzymes, which are purified from bacteria, and it uses chemicals like nucleotides and cap analog. So the whole production of messenger RNA in vitro is vegan. It's not the case for viruses. As I told you earlier, for the adenovirus of AstraZeneca, it's produced on human embryonic kidney cells in cell culture, which normally also use animal serum. So for some problems, for some people, it's a problem, and this is kind of solved with IVT mRNA vaccines because they are produced in a vegan conditions without any product from animal origin. And finally, about the production, to produce 1 million of doses of mRNA vaccine, you need 6 liters of in vitro transcription. So you can do only in 6 liters, if you do an in vitro transcription in 6 liters, you will have at the end enough RNA to make 1 million of mRNA vaccine doses. Usually when you do viruses, adenoviruses or other kind of viruses, by cell culture, you need 5,000 liters of cell culture to produce enough virus for 1 million of doses. So that's also an advantage of the mRNA vaccine. You can produce many doses in a small volume, while normally for the other type of vaccines that exist since many years, you need very large installations that contains thousands of liters to produce millions of doses. Are there some questions on this?
Yes,
unexpected aspects?
maybe we can answer some questions. So Sarah is asking, is vaccination recommended for people under antidepressants?
This I don't know.
Okay.
I would say that there is no reason to exclude them from vaccination. So as far as I know, such patients get the usual vaccinations against influenza or yellow fever or whatever. So I don't think that they should be excluded from the vaccination campaign. But again, the physician treating the patient will know better what to do because each patient is different and the file has to be checked carefully.
So the other question is concerning arthritis. Is it recommended for persons with arthritis?
Again, in autoimmune disease, autoimmune disease normally are not an exclusion criteria for vaccination. People with autoimmune disease, lupus, rheumatoid arthritis, etc., they still get vaccinations normally. There are exceptions. Again, the physician will know if there are reasons to exclude one autoimmune patient from getting a vaccine. But generally speaking, autoimmune patients do get vaccinated.
Do you recommend taking aspirin after vaccination to avoid any thrombosis?
No, I think there is no reason to think that the vaccines give thrombosis, at least not the mRNA vaccines. The AstraZeneca vaccine is being checked for the risk of thrombosis, but we will see what AstraZeneca decides. If they suggest to take aspirin after the vaccination, I would doubt it. So in general, I think this risk of thrombosis is just the normal risk in the population of getting thrombosis. Again, thrombosis happen every day, everywhere, with or without vaccines, something which is
Roy is asking his question in French. Est-ce qu'il y a des risques de recombinaison, de lien avec l'ADN du corps?
Oui, ça c'est ce que j'ai dit tout à l'heure. Donc là, c'est vraiment un des risques théoriques, recombinaison de l'adénovirus d'AstraZeneca ou Sputnik V ou Johnson & Johnson qui recombine avec d'autres adénovirus ou bien qu'ils s'intègrent dans le génome. Lorsque vous injectez un adénovirus dans une souris en intraveineux, vous trouvez après dans le foie une cellule sur 1 million qui a intégré dans son génome l'ADN, une partie de l'ADN de l'adénovirus. Donc c'est quelque chose de naturel qui se passe naturellement. Les adénovirus sont des virus à ADN. Leur ADN, lorsqu'ils infectent une cellule, va dans le noyau de la cellule et parfois ils s'intègrent. Comme je le disais, dans la souris, dans le foie, une cellule sur 1 million, dans une souris chez qui on a donné de l'adénovirus, contient dans ses chromosomes de l'ADN d'adénovirus. Donc c'est quelque chose de naturel qui va se passer dans les campagnes de vaccination. Donc là, il y aura donc chez les gens injectés par le AstraZeneca, le Sputnik V, le Johnson & Johnson, il y aura probablement donc des intégrations parce que c'est quelque chose de naturel qui se passe avec les virus à ADN de toute façon. Alors, est-ce que la fréquence sera de 1 sur 1 million ou de 1 sur 100 millions? Je ne sais pas, mais naturellement, ça se passe. C'est quelque chose de documenté. Les adénovirus, parfois, s'intègrent dans les chromosomes. Ça sera probablement le cas pour ces adénovirus déficients. Il n'y a pas de raison de penser qu'ils ne suivront pas les règles normales d'intégration qui arrivent pour les adénovirus. Et c'est là où j'ai un problème théorique, à nouveau, c'est qu'effectivement, il y aura peut-être des cas d'intégration de ces adénovirus vaccinaux chez certaines personnes et avec un risque potentiel dans des années, dans 5 ans, 10 ans, de développer une transformation initiée par l'intégration de l'adénovirus. La cellule HEK, Human Embryonic Kidney, dont je parlais tout à l'heure, qui est utilisée pour produire le vaccin, est une cellule transformée par adénovirus. Donc les adénovirus sont connus pour transformer parfois les cellules. Donc à nouveau, on est infecté par des adénovirus durant l'hiver. Donc on a tous été exposés à des adénovirus, à des ADN d'adénovirus, et on n'a pas eu de transformation. Donc ce sont des risques théoriques, des choses très peu fréquentes. Mais vu que les constructions dans les adénovirus d'AstraZeneca, Sputnik V et Johnson & Johnson sont des nouvelles constructions, on n'a pas de recul sur la fréquence d'intégration, sur la persistance.
Voilà, il y a Joël qui dit pourquoi le vaccin Sputnik n'est pas recommandé, mais je pense que vous avez répondu, parce que le Sputnik, il fait partie des adénovirus, non?
Oui, le Sputnik, l'AstraZeneca et le Johnson & Johnson sont tous les trois des adénovirus recombinants déficients.
Il y a donc ce risque de recombinaison.
Pour ces trois-là, voilà. Le risque théorique.
What about COVID-19 vaccines during pregnancy?
There are some ongoing clinical studies. That's a difficult question. You may have heard that there seems to be a higher rate of pregnancies that were stopped because of a SARS-CoV-2 infection. So it looks like the SARS-CoV-2 virus could have bad impact on pregnancy. So women who are or who want to be pregnant should be vaccinated probably, but of course we don't know about the vaccine in this person. So there are some ongoing clinical studies from Pfizer-BioNTech, for example, where they vaccinate pregnant women in order to see eventually or to show that it's safe. It's difficult because if a pregnant woman gets the infection, there is a risk for the pregnancy. So it's important to know if pregnant women could be safely vaccinated. In theory, yes. Practically, it's being tested in clinical studies.
Okay, maybe we can take one more question. Marcel, does the attenuated vaccine can positive the RT-PCR for COVID-19?
No. So the PCR for COVID-19, they are testing different parts of the SARS-CoV-2 genome. In the vaccine, whether the recombinant vaccine from AstraZeneca, Johnson & Johnson and Sputnik V, or from the mRNA vaccine, has only one small part, the spike of the SARS-CoV. So the PCR will detect only the spike and not the other part, so it would be negative.
Okay. What about long-term immunity for mRNA vaccines? Do we have any data about the term immunity, the long-term immunity?
No, so these formulations are new. The formulation of synthetic mRNA in this liposome from Moderna, BioNTech, or CureVac given intramuscular. So they are used only since 1 year. So the companies are taking blood from the healthy volunteers who have participated in the clinical study starting in March 2020. So we have now at best 1 year from the first person who has volunteered for the Moderna vaccine. It was 16th of March 2020, so 1 year ago exactly. So in this one person, you can test in her serum how much antibody is existing. So we will learn with time in these very early volunteers that are very courageous and really brave. We can follow them and see in their serum how the antibodies are persisting or going down. For the time being, there is not, of course, more than 1 year exactly of overview.
Yes. Maybe we can continue the presentation and then answer others' questions.
So I will go to some timelines about the mRNA vaccines because people say it's new, but of course, as I said, first, there are other types of mRNA vaccine like yellow fever that have 60 years of experience. But when we speak about synthetic in vitro transcribed mRNA vaccines, then the expertise has started in 1990 when a team of the US has published that in vitro transcribed mRNA made in vitro, when injected in a mouse, can give protein expression. So this was the first demonstration here in 1990 that in vitro transcribed mRNA can give in vivo expression of a protein encoded by the mRNA. 3 years later, a French team from Professor Meulien has shown that mRNA produced in vitro encoding for influenza protein could be formulated in a liposome and given to mice, and the mice developed immunity against influenza. So this is very important because this was the first ever demonstration of the mRNA vaccine. It's very similar to the formulation that are used now, 30 years later. It's in vitro transcribed mRNA in a liposome, given here in mice and showing that it is working as a vaccine. So this here was the first time people could demonstrate the EIVT mRNA vaccination. 2 years later, another team in the US has shown the same, but with naked mRNA and mRNA coding for cancer protein, and they could show that this gives indirectly an immune response against cancer. And then in 2000, the head of the immunology department in Tübingen, Professor Ramensee, has published with Igmar Her, who was his PhD student, and also Professor Gunther Jung, that mRNA naked or in a polyplex, so not in a liposome as was done before, but in a polyplex, could also be used for vaccinate. So this project of using mRNA for vaccination was an idea of Professor Hans-Georg Ramensee in 1996, and he has given this project to Igmar Her, and Igmar Her has developed, has optimized the formulation of protamine and naked mRNA. And when I joined in 1998, the department of Professor Ramensee, I got to know Igmar and I got to test and further optimize, I'm here, further optimize the mRNA vaccines. And we have created then in 2000 CureVac together with Florian von der Mulbe. And the idea here was not only to do science, but really to bring the technology to patients, to the hospital. We were mostly focused on cancer vaccines because Professor Ramensee worked already since many years on cancer vaccine using peptides for vaccination, not mRNA, but he still had the idea of using the mRNA. And so the idea of to create the company was really to go to the clinic. And one of the first steps was the production of mRNA in pharmaceutical condition. The mRNA you produce in the laboratory, you can use it in vitro and for animals, but you cannot use it, of course, for humans. to use it for human, you need to produce it in clean rooms. You know, this kind of room with very clean air where you enter with a kind of costume, where you are very protected, where every step is monitored and checked and everything is perfect. So this is called clean room. And the production of a drug has to be done in clean room. In 2000, we went to several companies in Europe producing, for example, DNA for clinical studies, but they didn't want to produce mRNA for us. They didn't have a process and there was no market. They were not interested. They were considering that we are a bit crazy to do mRNA-based vaccines. So we had to do it on our own. So we had to find money, investors, in order to establish clean rooms, hire people, and really put in place a pharmaceutical production, which is unusual for a small biotech company, but we had to do it because mRNA did not exist. We created the company, we got a little bit of publicity in the newspaper in 2001. It was very local because at that time, nobody was believing in the mRNA technology, in the mRNA vaccine. I can tell you, I'm very happy now to have questions because when I was presenting our data in 2000, 2001, 2002, I never had any questions in congresses. So I knew that at the end of my talk, when the moderator asked, are there some questions? Nobody would raise their hand and it would be this embarrassing moment where you think, okay, nobody cared. I was so used to it that I was preparing my own question at the end of the talk. So I was usually, for example, not speaking about the production. And when the moderator was asking their questions, I was myself asking me my own question, telling, you may want to know how we produce, and here is how we do it. So because nobody was interested in mRNA vaccine at this time, and it was worse than that. People from the scientific and medical community were thinking we're a bit crazy and that will lead nowhere. And this remained until 2019. I still had in Zurich colleagues telling me that the mRNA vaccine will never be useful for any kind of medical purposes. Of course, 2020 has changed everything. But at that time, in 20 up to 2015, there was barely anyone in the world thinking that mRNA vaccines would be useful, apart from the people in CureVac, in BioNTech, and in Moderna. So one of the first steps, as I said, was to produce messenger RNA in pharmaceutical conditions. Once we had it, we could inject it in human, starting with me here, because the idea, I wanted to check that the functionality of in vitro transcribed mRNA can be seen in human, and it's not only in mice. Theoretically, you could think that only mice can somehow take up the mRNA that you inject and express it. So I wanted to see if human can also do it. So I produced in pharmaceutical condition with my team in CureVac the luciferase coding mRNA. So this is the mRNA coding for the protein luciferase, which the firefly use in order to make light. So I got here injection in my skin of mRNA coding for luciferase, and this is the trace of the bubble that makes the intradermal injection. And one day later, under local anesthesia, we made punch biopsies. So we took pieces of my skin at a few places, at the place of injection of the mRNA or at other places. And then we measured in these biopsies emission of light. And we could see that biopsies taken at the place of mRNA injection, mRNA coding for luciferase, emitted light, which showed that the human skin, like the mouse skin, if you inject in vitro transcribed mRNA in it, it can take up the mRNA and express the protein. So this was a step towards clinical studies to show that in human, in vitro transcribed mRNA is expressed. We did a few clinical studies that we published starting in 2008, vaccination of melanoma patients and renal cell carcinoma patients. And at the same time, at the time of our first publication, which appeared at the really low impact factor, because again, at that time, nobody was thinking that our work would have an impact, and we could not publish in a high impact factor journals. We had to go, I submitted to Nature, but it was immediately rejected, and then to many other journals, and ending up to impact factor 3, because reviewers were all telling us, we do crazy things, and that's of no interest. So we published in 2008. I will show it on the next slide. In 2008, Until 2008, CureVac was the only company dedicated to mRNA vaccine, but in 2008, Professor Ugur Sahin and Özlem Türeci have created BioNTech in Mainz. So it's a university of Germany. So CureVac was in Tübingen and BioNTech in Mainz is 200 kilometers away. And in 2010, in Boston was created Moderna. So in 2010, we had in the world 3 companies focused on using mRNA for vaccine and therapies, Moderna, BioNTech, and CureVac. So this has made a big change because there were more people thinking and seeing the potential of mRNA vaccines and joining us. And in particular, the people in BioNTech who are particularly good and wonderful and who have made a big change in the development of mRNA vaccine. As I show here, these are the first 3 publications on mRNA vaccine, the first ever publication on mRNA vaccine in cancer patients. My injection, so the injection in my legs, we published in 2007, but these were only injections to monitor expression. The first clinical study where we monitored immune response after injection of mRNA vaccine is in the title. It was published in 2008 in Journal of Immunotherapy, impact factor 3.5. And a second trial we have published in 2009. And this was also in Journal of Immunotherapy. It was again melanoma patient, but it was not naked RNA. It was polyplex mRNA, so no liposome, but a polyplex formulation. And another trial we did in cancer, in renal cell carcinoma patients, and this we published in 2011. So as you see, these are all low impact factor journals because again, nobody was interested. It has been very different with the work of BioNTech. BioNTech has done a great work developing, optimizing the mRNA vaccine. BioNTech is Pfizer for the COVID vaccine. So BioNTech has improved the mRNA vaccine, and they could show in several articles in Nature that when they inject mRNA in cancer patients, they get a strong immune response against cancer. And this publication was the first one in 2016, showing a liposomal formulation of mRNA in cancer patients, showing how safe and efficient it is to induce an immune response. And then Professor Ugur Sahin has developed further some personalized RNA vaccines and showing how efficacious they are. And this work is ongoing, mRNA vaccination in cancer patients, which is very promising for the treatment of cancer or prevention of cancer. Any question on the history of mRNA? If not,
yes,
I will
we have
move
some questions. There is a question related to cancer. Actually, Diana is asking, what about vaccines against cancers? So
yes, I heard the first answers of Dr. I don't have, but I would like to know all kinds of cancer profit from this vaccine, from this kind of vaccines.
Yes, that's the power of this approach. Because mRNA is so easy to produce, now what BioNTech is doing is personalized RNA vaccines. In this approach, we take a biopsy of the cancer, we sequence the genome, and we find the mutations compared to healthy tissue. And then we design an mRNA vaccine that triggers an immune response against the mutation. Every patient gets his own vaccine. It takes 3 months between the biopsy and the vaccine. It's called individualized vaccines. So it's very important because it doesn't depend on the type of cancer. In theory, whether it's a mammary cancer or leukemia or other type of cancer, as long as you have a biopsy, you can find the mutations and make an mRNA vaccine that codes for the mutation and that will trigger the immune response against the mutation of the precise cancer in one person. And the clinical studies that are ongoing from BioNTech, they do recruit many types of cancer patients. most of the several of the clinical studies are not specific of one cancer. Some are melanoma, but some are only solid cancer. Any type of solid cancer can be pancreatic cancer, lung cancer, mammary breast cancer. Any type of solid cancer is recruited in these clinical studies of individualized RNA vaccination.
So it is still in study?
It's in clinical study, yes. In phase 2, it was showing some efficacy as a monotherapy, but not enough. So what is being done now is to combine this vaccination with standard chemotherapy or radiotherapy or immunotherapy as combination.
Yes. And since which year it was talking about?
You mean when it will be approved?
It's not approved yet.
No, it's not approved yet. It's in clinical development, in clinical studies. And in cancer, the clinical studies, they take a long time because most of the time you want to see if the vaccine, if the treatment has an impact on the survival of the people. So you need quite many months and years to show efficacy. It's not the case when you vaccinate against a viral disease like in COVID, because in the COVID, you can quickly see whether your vaccine is efficacious or not. In cancer patients, you need to wait 2 years, 3 years to see if the vaccine has given patients an advantage in surviving.
And is the vaccine efficient for people already sick of cancer?
Yes, the clinical, again, I cannot say it's efficient, it's in clinical studies, but all these clinical studies, they recruit patients who had previous treatments and who failed. So the clinical studies are open to patients who have failed first-line treatment in some cases, so that can then get recruited in clinical studies from BioNTech, where the individualized mRNA therapies are being tested. But it's clinical
studies. And for
studies.
people who had already cancer and that they are in remission?
No, if someone is cancer-free and has recovered, I don't think there are clinical studies in patients who have recovered from cancer.
Okay.
Okay, thank you. Rana is asking, is there any long-term side effect for Pfizer vaccine?
For the COVID, you mean?
Yes.
Not as far as I know. So the BioNTech Pfizer vaccine has started in April 2020. So we have less than 1 year overview. So as far as I know, there was no long-term side effect. There are short-term side effects in some people, sometimes more, sometimes less, like for every vaccine. I didn't hear about a long-term side effect. You know, when you vaccinate 100 million of people, you will always find people that 1 week, 1 month after the vaccination will have an issue. But probably they would have had the issue anyway. It's very difficult if you have very rare cases of people having, as we spoke about embolies, for example. Embolies happen every day in many people. So if it happens in someone who was vaccinated, it's very difficult to correlate to the vaccination. It takes time, you need many cases. Now that the mRNA vaccine from Pfizer BioNTech was injected in many millions of people, I think we can really say that it's safe.
Sarah is asking, does getting the Pfizer vaccine while having COVID-19 had adverse causes? Can it cause death?
No, it looks like it helps people. There are some people who have this long COVID, so who are sick from the SARS-CoV-2 infection during many weeks. And it looked like in these people, when they get vaccinated, they get quickly healed from the SARS-CoV-2. So it looks like vaccinating people who are having this long COVID helps them to recover.
Okay. Rabia is asking, adenovirus vaccines have been used for a long time now. Does the theoretical concerns have been detected on humans? And there is no problem with the mRNA modifying our genome?
No, but I think this is wrong. Recombinant adenoviruses have the first recombinant adenovirus vaccine has been approved in July 2020. And it's a vaccine protecting against Ebola. So there is no recombinant, I say recombinant adenovirus vaccine. The first one approved was 2020 against Ebola. So it's a new technology. It's actually studied since over 40 years, but the first approved vaccine based on recombinant adenovirus was July 2020 against Ebola. It's a new technology.
Yes, maybe we can continue and then we can address some more questions later on.
Sure.
Thank you, Steve.
So I have only two more slides. So we will focus on the
and
results.
Yes.
We focus on COVID and on the mRNA vaccines against COVID. So as I said, there are three companies, Moderna, BioNTech, and CureVac, leaders in the mRNA vaccines. Moderna was the first one to start with the injection of the healthy volunteer on March 16. BioNTech started the injection of healthy volunteers on April 23rd, 2020, and CureVac started a bit later. So all three companies, they use mRNA coding for a full-length spike in the prefusion stabilized form. So the PP modified, the proline modified spike for all of them. Moderna had a range in the phase 1 of 25 microgram up to 250 microgram of mRNA in the liposome, given intramuscular for all the companies. There were like 15 participants in each group in the phase 1. And they could decide for 100 microgram to go in the phase 2, phase 3, and this was approved 100 microgram. Pfizer, BioNTech, they had 4 different mRNA vaccines. So in parallel, that they tested in April. So not only one, but 4 different. One mRNA was coding for the full-length spike protein modified, PP, and this mRNA was either modified pseudouridine or non-modified uracil. As I said, there is no reason to think, even so far, that modified is better or less good than non-modified mRNA. So BioNTech, the expert in this field of modification, has tested in healthy volunteers both versions of the mRNA vaccine, non-modified and modified. Later, they have decided to go with a modified for the approval, but they did not yet publish the results of the non-modified. Here in the BioNTech study, it's the first time that there is a comparison between modified and non-modified mRNA in a vaccine. So when they will publish the non-modified data, we will know for the first time whether the modification of the mRNA is useful for a vaccine or not. So on top of these 2 versions, modified, non-modified of the full-length mRNA, they had a shorter mRNA coding for the receptor binding domain, and they had also a replicating mRNA. It's another technology I didn't speak about, but the results are not published. This technology requires less mRNA, and I'm curious to see the data that they obtain, but this is not yet published. So BioNTech has started in April 23. Their dose range in the phase 1 was broader than Moderna. They used from 1 microgram up to 100 microgram of mRNA per dose. And then they decided to use 30 microgram for the phase 3 and for the approval. So now we have 2 approved vaccines, the Moderna at 100 microgram mRNA per dose, and the BioNTech at 30 microgram per dose, BioNTech Pfizer. The CureVac vaccine in phase 1 was tested from 2 microgram per injection to 12 microgram. It's a non-modified mRNA, so it's used at a lower dose than the modified mRNA. And the ongoing phase 3 uses 12 micrograms. So if this vaccine will be approved, it will be a lower dose than the BioNTech and Moderna vaccines. I show an example of the immune response that was detected in the healthy volunteers that have agreed to be cobayes to test the vaccine. So here we look at the blue bars. The blue bars are the mRNA from BioNTech Pfizer coding for the full-length spike. And the red ones are the shorter version of the mRNA, which was stopped. So here we look at the blue bars. You have two cohorts of healthy volunteers, the young ones, 18 to 55 years old, and elderly, 65 to 85 years old, and three different doses of the mRNA. This was a phase 1 dose escalation, 10 microgram, 20 and 30 microgram. And you can see here at day 21, 3 weeks after the first injection, many of the healthy volunteers had neutralizing antibody titers, quite similar here to the people who have recovered from SARS-CoV-2. These are sera from patients who have recovered from the disease and they have neutralizing anticorpus in their blood. These titers you see here. So with the vaccination, after 1 dose, there was already some neutralizing antibodies, and after 2 doses, so 1 week after the second dose, the neutralizing antibodies you see here at every dose, 10 microgram, 20 or 30, were giving even higher antibody titers than the infection itself, showing that here the vaccine is very efficacious, and this was true in the young people as well as in the elderly. You see here at 20 microgram or 30 microgram of vaccine. After the second dose, all healthy volunteers have high levels of neutralizing antibodies against SARS-CoV-2. So this shows you how amazingly efficacious is this vaccine. With 30 microgram injected twice, you get antibodies that can neutralize the virus even better than people who have been infected and who have recovered from the virus. My final slide summarizes the 3 mRNA vaccines, the 2 that are approved, Moderna, BioNTech, Pfizer, and CureVac. We are waiting for the results in March. So again, Moderna is modified mRNA, 100 microgram. BioNTech is modified mRNA, 30 microgram. CureVac is non-modified mRNA, 12 microgram. And the results from BioNTech Pfizer were confirmed in Israel because Israel has vaccinated more than 50% of its population, of its 8 million people in Israel, and half of them at least have received injection of the BioNTech Pfizer mRNA vaccine. And they could show indeed that the vaccine protects, is efficacious at 97%, which is what BioNTech Pfizer has published from the phase 3 study. They published 95% efficacy. In Israel, on large population, they can see 97% efficacy against COVID. And importantly, they find 94% efficacy against infection, showing that the vaccine does not protect only against the disease, the COVID, the symptomatic disease, but it also protects against infection. So the drop of 94% in positive PCR positive tests was seen in vaccinated people 1 week after the boost. Another study has compared the Moderna and the BioNTech Pfizer vaccine and shows that the BioNTech Pfizer vaccine is slightly safer, has less side effect than the Moderna. It could be because of the dose. Moderna has 100 microgram and BioNTech Pfizer has 30 microgram. Both are safe, but this study here, the recent study, tend to show that there are less side effects with the BioNTech Pfizer vaccine. And we are waiting for the CureVac results. This is the end of my talk. If you want to know more about mRNA, you can go on our platform, or you can look at this article that I published in February 2021 in the journal Viruses. You can contact me if you have more questions, and I'm happy also now to answer your questions.
Thank you so much. Thank you so much, Professor Pasculo, for this amazing webinar. So we still have like 15 minutes for questions. Do you want to stop sharing the screen so we can discuss with everyone? You can also open your cam if you want. You have like 15 minutes for discussion. So I will start by the last question. To be protected against SARS-CoV-2, are we supposed to be vaccinated on a yearly basis? If yes, do we have to take a different vaccine each year?
This we don't know. The SARS-CoV-2 is 1 year old, so we cannot go faster than the time. We have 1 year overview. As I said, the first volunteer was injected on the 16th of March. So we don't have more than that to know how long is the immune response and whether we will need new injections. The problems could be the variants. So far, as I said, the mRNA vaccine from Moderna and from BioNTech Pfizer induce antibodies which protect against all variants, including the variant identified in South Africa. The AstraZeneca vaccine does not protect efficiently against the variant from South Africa. So, so far the mRNA vaccine, they protect against all variants. So in theory, people who are vaccinated should be resisting to SARS-CoV infection so far. But it cannot be excluded that we get a SARS-CoV-3 or that we get a variant which escapes from the vaccine. That's a possibility. In that case, the mRNA vaccine will be produced again to code for the new spike protein, which will trigger an immune response that will protect against eventual new variants. But so far, it's not needed. The BioNTech, Pfizer, and Moderna vaccines, they give antibodies that recognize all variants that have been tested.
Yes, Maria is asking, do you recommend vaccination for young people without health problems?
Yes, if there are enough doses, yes, it should go first to people who are fragile. So I would not give a dose to someone young and healthy if he has himself no risk and if he's not in contact with somebody who has a risk. So I think at the moment we still have not so many doses available in our countries, so they should be prioritized to be given to people who are fragile first. Once the population with fragile is protected and also the medical people, then after that we can think about vaccinating other people. In Israel, where they have vaccinated all fragile people, they start now to vaccinate people from 16 years old. So in Israel, they are starting to vaccinate young people who have no risk normally to develop COVID, but to vaccinate them prevents the transfer and the infection to other people. So Israel is starting to vaccinate 16 years old.
Okay. Isra is asking, do we have experimental evidence that the injected synthetic mRNA will be degraded in short time?
No, so we do have. I mean, when you inject mRNA coding for example, luciferase in animals, you can follow the kinetic of expression. And whether you use mRNA in cell lines in vitro or in vivo in animals, you always see expression coming after a few hours, being maximal, and then going away. And it can go away within 1 day in cell culture because the cells divide and then they lose the mRNA. In vivo, it can stay a few days. People have used very stable mRNA coding for erythropoietin with the idea to get a long-term expression of erythropoietin. And you can find the erythropoietin a few days after injection of mRNA coding for erythropoietin. So it's possible to have a few days of expression, but after 1 week, you never see anymore any protein coded by the mRNA, meaning the mRNA is gone.
Okay. Yumna is asking, if we got vaccinated against COVID-19, can we still catch and spread the virus?
In theory, yes. You know, it's not black and white in nature. Of course, if you get a lot of virus coming from someone who is heavily infected, and then even if you are vaccinated, you will get a high dose of virus, I would say, and some of it could eventually a little bit replicate during 1 or 2 days before being neutralized. What they see in Israel, as I just showed, is 94% drop in the PCR positive test in the population that is vaccinated, showing that people vaccinated are more negative in PCR than people who are not vaccinated. So that shows that it reduces infection rate. So that's clear now in Israel. It could not be studied in the clinical studies in phase 1, 2, or 3 because it involves too many people. But in Israel, because more than 50% of the population could be vaccinated, now they can see that the rate of infection goes down, meaning the vaccine protects indeed against infection. And in monkeys, the studies that were done with this vaccine in monkeys have also indicated that in vaccinating monkeys, if you give them the virus in the nose, after 3 days, you don't detect any traces of the virus, while in non-vaccinated monkeys, after 3 days, you still detect a lot of the virus in the nose and in the lungs. So that was also showing in the monkeys that when you vaccinate and you give a high dose of virus, after 3 days, it's all gone, while without vaccination, after 3 days, it's ongoing replication.
Thank you, Steve. One more question. Do COVID vaccines cause autoimmune diseases?
If COVID vaccine could trigger autoimmune disease.
Autoimmune diseases.
The question is whether the vaccine could trigger an autoimmune disease.
Yes, yes.
Yeah. That's a theoretical concern for every vaccine. When you get your measles, mumps, rubella vaccine, when you get your hepatitis B vaccine or HPV vaccine, you always activate an immune response. And it's hard to know in some rare cases of autoimmune patients whether the autoimmunity has any link to the vaccination. You know, for example, for multiple sclerosis, the people have a statistic risk of multiple sclerosis of the country where they live. So immigrants coming to a country, they get the risk of developing multiple sclerosis in their life, which is normally the risk of the country, showing that there are some environmental impacts on the risk of developing autoimmune disease. So far, it was not clearly found what are these environmental impacts. Vaccination could be one, because depending on the country, you don't get the same vaccines. So in theory, it could be, you may know that in the 90s, there was a big discussion about the hepatitis B vaccine and multiple sclerosis. Some people have claimed that they got multiple sclerosis after being vaccinated. It was checked, and there was no relationship that could be identified between hepatitis B vaccination and the rate of multiple sclerosis. So, so far, there was never, as far as I know, any proof that a vaccine, whether influenza, hepatitis B, yellow fever, would trigger some autoimmune disease, but it's a theoretical concern for every vaccine. When you stimulate the immune response, you cannot exclude that in some people, after 10 years, 20 years, somehow the triggered immune response will cross-react with something else. But the risk is present from what we breathe, from what we eat. You have some immune responses from viruses you meet in your life, and we don't know so far what triggers autoimmune disease in autoimmune patients.
If you were to be vaccinated, which one you will choose? It's very, I don't know if you can answer this question, but what I will ask you, please, if you can open your cam now, and then ask directly your questions, and then you can take a photo souvenir for our website of this webinar. So may I ask you to open your cam if it's possible, please? Thank you very much. If you can open your cam, thank you very much. So we'll take a photo. We have just 3 minutes, more minutes. If you have questions, please add them directly to Steve. Do you have more questions to ask directly to Steve?
J'ai envoyé 2 questions.
Tu veux les poser? Vas-y, Diana.
Je voulais savoir s'il y avait un seul traitement pour le corona qui soit efficace pour tous les cas.
Contre le virus, que je sache, il n'y a pas vraiment pour l'instant de traitement établi contre l'infection. Par contre, contre la maladie, contre le Covid-19, les stéroïdes sont vraiment une des clés qui permet de mieux traiter la maladie. L'infection, à nouveau, je ne connais pas de médicament qui soit approuvé contre l'infection. Il y a eu beaucoup de discussions sur différents médicaments. L'ivermectine semble prometteuse contre le SARS-CoV, contre le virus lui-même, mais que je sache, ça n'est pas encore approuvé. Par contre, contre la maladie, le Covid, Donc là, vraiment, ce qui a changé les choses, c'est le traitement par corticostéroïdes des patients au moment où les patients doivent être ventilés. Le traitement de l'inflammation permet de sauver beaucoup de vies. Donc, à votre question, je crois que pour l'instant, il n'y a pas de médicament qui empêche la réplication du virus, qui soit approuvé. Par contre, le traitement de la maladie, lui, est bien meilleur grâce à l'utilisation d'anti-inflammatoires.
Merci.
Entre-temps, Nathalie va mettre un petit questionnaire pour feedback. It's a small assessment grid. If you can answer the questionnaire, it's on the chat box. So more questions in French, in Arabic, in English. The language is not a barrier.
Do not hesitate to ask questions of things you did not understand, because that can help me next time to explain better.
So more questions? On peut avoir accès à la présentation. We'll be sending you the recording. The recording, Steve agreed that we send you the recording, so you will receive the recording of this webinar.
Wonderful.
So do you have more questions? Pas de questions?
En fait, Fad, il y a une question dans le chat box. Do you agree when we say that any vaccine is better than no vaccine, even
Yes.
for people who are not frontline workers?
Yes,
Okay.
yes. At this stage, every vaccine, at least that have gone through the testing in Western world, it's a little bit harder to evaluate the vaccines from China, the inactivated SARS-CoV-2, because we don't have the data. But every vaccine that has been approved in Western world, so the Moderna, the BioNTech Pfizer, the AstraZeneca, the Johnson & Johnson, so all these vaccines are efficacious and safe in general. So they should be, if you can be vaccinated by any of them, I would say I would recommend to get vaccinated. That's the way to get over with the pandemic. And Israel shows the results that they have vaccinating one population. They finish the lockdown, so they reopen everything now. Life is going as before. They have very low number of deaths and number of sick people. So by vaccinating their population massively, they are out of the pandemic, the first country in the world. So that really shows that if you vaccinate, you can get out of the pandemic. So I think our countries have suffered from death and disease and also social and economical impact from the pandemic. So yes, every vaccine that is now available should be used. And I think everybody could participate in ending the pandemic by getting vaccinated. I recommend, especially for young, healthy people, to get the mRNA vaccines because there is no long-term side effects. So in general, of course, I recommend the mRNA vaccine. What I didn't say is that in 1998, when I started, the mRNA vaccine was not efficacious in animals compared to DNA vaccine or virus vaccines or protein vaccines. So the whole history of mRNA vaccine is safety. In 1998, I thought, okay, we need to optimize it because it's not working well. It does work a bit, but not well. But because it has this safety features, because it does not persist, because it's degraded, that's a wonderful format to be very safe. So the whole history of mRNA vaccine, at least as far as I'm concerned, since 1998, is to develop this format because of safety. At that time, we had DNA vaccine, we had other type of vaccines, which I thought were not safe enough, especially DNA vaccine, because of the problem of potentially persisting and recombining. So the development of the mRNA vaccine all came from safety first. And it was not optimal, but with the years, with the work of mostly the companies, the efficacy of the vaccine could be really now wonderful and high, but the whole history of mRNA vaccine was safety first. So mRNA vaccine are super safe vaccine, and that's the reason why they have been developed.
The last question, I know you are tired, Steve, so just take one more question. Heidi is asking, why did Sanofi surrender so quickly and stop developing a vaccine?
I think that they worked on, Sanofi is an expert of protein vaccine. And as I have showed in my slide with a smiley, to make a protein vaccine is not easy. If you are a company expert in doing a protein vaccine against hepatitis B, and you have to do a protein vaccine against SARS-CoV, you have to start from zero, because the way to produce a protein A and protein B is very different. You have to re-optimize the condition of production, you have to re-optimize the condition of purification, of preservation, which adjuvant you use. So for each new protein, you start from zero. So even if you're an expert of protein vaccine, it doesn't mean that you can do a new protein vaccine against a new virus. And that's different with mRNA. If you're an expert in mRNA vaccine, you can easily produce mRNA vaccine against any virus because the process is the same, whatever is the mRNA you produce. But when you produce protein, it's very difficult. As far as I know, Sanofi has tried to produce the spike protein. It's a big protein. It's glycosylated. It's difficult to produce. And as I showed with the pre-fusion conformation, this protein can have different conformation. So if you produce a protein that has the wrong conformation and you vaccinate with that, you will get wrong antibodies. So it's very difficult to work with proteins. So Sanofi being great in protein vaccine, they have great vaccines that they have optimized during many years against viruses like hepatitis B. But when you come with a new protein, they have to start from zero. And as far as I know, they had issues with the protein production and the antibodies triggered by the protein.
Un grand merci, Steve, vraiment pour ce webinaire passionnant. Et on a eu plein d'informations. Il y a Diana qui a encore une question avant. Oui.
C'est-à-dire qu'il y a quelqu'un qui a posé une question intéressante. Je ne sais pas si Steve peut répondre. Est-ce que vous prévoyez la fin du corona à un certain moment?
Oui, donc à nouveau, on voit en Israël que l'épidémie diminue. Maintenant, il semble que notre manière de vivre dans le monde soit propice au développement de nouveaux virus. On a eu donc le SARS-CoV-1 en 2002, le MERS en 2012. Il y a malheureusement donc entre les animaux et les hommes des interactions qui ne sont pas forcément très naturelles, donc dans des marchés où des gens vont vendre des chauves-souris vivantes et les mettre dans des cages avec des lapins et des poules. Donc il y a dans l'activité humaine malheureusement de nos jours un potentiel de créer des nouveaux virus en mettant ensemble des espèces qui ne se retrouvent pas ensemble normalement ou en les mangeant. Et donc, ça crée un monde où il y a des virus qui passent d'une espèce à une autre de manière assez rapide et une fois qu'ils infectent l'homme, peuvent être distribués dans le monde entier assez rapidement. Donc, il est probable que notre monde soit propice à des épidémies régulières qu'on n'a pas eu plus tôt, mais à nouveau, avec le coronavirus de 2002, de 2012, de 2020, on voit qu'il y a des risques d'épidémie, de réassortissement de nouveaux virus qui proviennent de notre mode de vie et de l'exploitation des animaux. Et donc, à mon avis, certainement, il faudra s'habituer à des pandémies, à des mesures contre les pandémies. Et donc, il est possible que l'année 2020 donc, soit une transition dans notre manière de vivre et nous rendre aussi plus vigilants et aussi, donc, effectivement, plus réactifs contre de nouvelles épidémies. Il y a toujours eu le problème de la peste porcine, d'autres potentielles maladies qui viennent des animaux. Donc, certainement, c'est un risque qu'on a dans nos civilisations actuelles de passage de virus d'un animal à l'homme et de transfert de ce virus à la population humaine par les déplacements. Mais on va sortir du Covid, comme Israël en est sorti maintenant, on va en sortir, mais certainement en gardant quand même des gestes barrières, en se vaccinant, en faisant attention aux variants, en étant très réactif contre les variants. S'il y a un SARS-CoV variant qui échappe à la vaccination, il faudra refaire des vaccins qui protégeront contre les nouveaux variants. Donc je pense qu'on va maintenant être très vigilant et se protéger et effectivement continuer à faire des vaccins, à prévoir des vaccins contre des variants potentiels de coronavirus ou d'autres virus qui pourraient affecter l'humanité.
Et vous travaillez encore pour d'autres vaccins en ce moment, Steve?
Moi, je travaille surtout sur des vaccins anti-cancer. Et effectivement, là, par le coronavirus, j'ai eu des soutiens de la Suisse pour développer un vaccin ARN contre le coronavirus. Donc, il y en a déjà. Mais l'idée, c'est d'avoir des vaccins qui fonctionnent avec encore moins d'ARN. Comme vous le voyez, donc le Moderna, c'est 100 microgrammes, le CureVac, c'est 12 microgrammes. Si on continue à améliorer la technologie, on pourrait avoir des vaccins qui fonctionnent avec 1 microgramme ou 0,1 microgramme d'ARN, ce qui permettrait de produire des doses beaucoup plus rapidement. Parce que si au lieu de 100 microgrammes, votre vaccin marche avec 0,1 microgramme, alors avec la même production, vous faites 1000 fois plus de doses. Donc les vaccins ARN fonctionnent, on peut toujours les optimiser de façon à les faire encore meilleurs, de façon à réduire la dose nécessaire par injection, de façon à produire encore plus vite et à distribuer encore plus vite les doses en cas de pandémie.
Et pour les malades de cancer, où est-ce qu'ils peuvent s'inscrire dans un essai qui leur permettrait d'obtenir un vaccin efficace? Auprès de qui?
BioNTech et Moderna. BioNTech et Moderna, si vous allez sur les pages web, ils ont un onglet pour les patients. Donc, vous pouvez regarder leurs essais cliniques, voir s'il y a des essais cliniques qui seraient peut-être relevants pour le patient en question. Et à ce moment-là, vous pouvez écrire un email et les compagnies ont des sous-contracteurs.
Et leurs essais cliniques sont par mutation génétique ou autrement classés?
BioNTech a plusieurs essais cliniques. BioNTech n'est pas une compagnie qui travaille que sur l'ARN messager. Ils ont des thérapies cellulaires, ils ont des anticorps. Moderna ne fait que de l'ARN messager, CureVac aussi, mais BioNTech a plusieurs plateformes de thérapie contre le cancer. Et donc, si vous regardez sur la page web, vous allez voir tous leurs essais anti-cancer, anti-mélanome, anti-cancer pancréas, etc., avec différentes stratégies. Ils ont des produits chimiques, ils ont des anticorps, ils ont de l'ARN, ils ont des vaccins, ils ont des thérapies cellulaires. Donc, ils sont très, très diversifiés. Et donc, les patients cancer qui souhaitent participer à des vaccins, à des essais cliniques anti-cancer, souvent trouveront chez BioNTech un essai clinique qui leur correspond. Moderna aussi a des essais cliniques un peu moins larges dans le cancer, mais ils en ont. Donc, si vous regardez sur les pages web de Moderna, de BioNTech, vous allez trouver donc les différents essais cliniques. Et à nouveau, ces compagnies ont un service, ils répondent aux patients qui leur écrivent, qui leur demandent s'ils peuvent être inclus dans des essais cliniques.
Merci.
Merci beaucoup Diana. En fait, Madame Diana Fadel, si vous ne la connaissez pas, c'est la fondatrice et la présidente de la Fondation Diane.
Félicitations à vous, bravo.
Dans ce cadre-là qu'on a monté la chaire et tout ce webinaire grâce à la proposition de Diana. Merci beaucoup à Nathalie et à Tracy. Grand merci au professeur Steve Pascolo. Et à bientôt, j'espère, dans le cadre de notre rencontre.
Merci à vous, merci pour l'invitation, merci à Diana, merci à vous tous.
Merci, au revoir.
Merci.
Bonne soirée. Et on va vous envoyer le recording. L'enregistrement vous sera envoyé avec l'accord du professeur Pascolo.
Merci beaucoup.
Bonne soirée.
Merci beaucoup.
Au revoir.
Bye bye. Au revoir.
C'est un plaisir.